Okay. Well, good afternoon, everybody, and welcome to the Pathology Grand Rounds. Today, it's my very distinct pleasure to introduce UAB's very own Dr. Barbara Gower. Dr. Gower started her training at Penn State, where she obtained her BS in microbiology before heading out west, where she completed her doctoral work at the University of Utah in Salt Lake City in biology. She then completed two separate postdoctoral fellowships, first at the University of Delaware, and then fortunately for us right here at UAB. And we were lucky enough to keep her here, where she's really flourished and is really a world leader in understanding nutrition science. Dr. Gower is now a full professor and vice chair for research in our Department of Nutrition Sciences, as well as serving as the director for UAB's Metabolism Core. She has a very impressive funding track record and current funding. She's currently PI on 3 NIH grants, 2 R1s and a P30, and co-investigator, co-PI on 3 other NIH grants. What really impressed me about seeing her full CV is her publication record. It's well over 265 publications and counting, many of them foundational in our understanding of diet and how it impacts diabetes and obesity. Her research really focuses on the interplay of those two areas, be it diet and the endocrinology system, and how it impacts metabolism. Her major research focus has been divided into 2 core themes. One really focused on this dietary intervention of low carbohydrate, low fat diets and how it can actually help with insulin function in type 2 diabetes. And the other one is really tackling this idea of personalized medicine, where she's been working to find the ideal diet composition for effective and sustained weight loss in African-American women who are often more at risk for this disease state. So I could go on and on, but I don't want to waste any more of her time, because today she's going to tell us about insulin resistance and obesity and the new sites that we've gained in those areas. Thank you, Dr. Gower.
Oh, thank you, Adam, for the kind invitation. And it's a pleasure to be here today. So the title of my talk is Insulin Resistance and Obesity, New Insights. And I've been, as Adam says, I've been in this field for over 20 years. And I feel like when I first got into the field, we had more questions than answers. And I think things have evolved a lot over the last 20 years. And that's what I'm going to talk about today, weaving in a lot of the data that we've collected here at UAB. So the learning objectives these are the 4, I've listed them here. Insulin resistance is genetic, but can be modulated by weight gain or weight loss. Greater insulin sensitivity is a risk factor for weight gain. Risk for type 2 diabetes in ethnic minorities has its roots in genes and physiology. Restricting dietary carbohydrates will reduce risk for type 2 diabetes, particularly in ethnic minorities. So when I first got into the obesity field, this is sort of what it looked like in terms of what we thought was going on. I should say obesity and chronic disease. Some people became obese, but we didn't really understand why. And then within that population, some people became viscerally obese. They developed that visceral fat depot, which we eventually learned was linked to chronic disease. But again, we didn't really understand why. Why did some people become viscerally obese, but others didn't? But nonetheless, once you had someone who was viscerally obese, they were insulin resistant, their inflammation was up, they were dyslipidemic, et cetera, et cetera, et cetera, et cetera, and they were at risk for chronic diseases, type 2 diabetes, cardiovascular disease. But what we now know is cancer is another one of these diseases, and even Alzheimer's has been thrown into this mix. They're just various manifestations of an underlying metabolic dysfunction. But what I'm going to show you today is that this scenario, which was never very satisfying in the first place, it's basically wrong. It's essentially it is wrong, and I'm going to show you what's wrong with it today, and I'm going to propose an alternative pathway. So the whole field has been evolving, like I said, for the past 20 years, and things have been, they've been sort of moving in a certain direction, and I feel that direction crystallized about 4 years ago when this paper was published. This paper was published in 2017. It's called Integrative Genomic Analysis Implicates Limited Peripheral Adipose Storage Capacity in the Pathogenesis of Human Insulin Resistance. And the study was done using 5 of the large European data sets. So these are the publicly available data sets that you can actually get, like the UK Biobank and so forth and so forth. There were over 300,000 participants in this study. And the goal of this particular paper or the study was to determine the genetic basis for insulin resistance. They did that by using fasting insulin as a surrogate for insulin resistance, but they adjusted for confounding factors and they validated it against the euglycemic clamp and the IVGTT. So it's fairly solid. And what they did is they had, SNP analyses, and they looked for genomic regions that were associated with this fasting insulin outcome. And they came up with 53 genomic regions that statistically were associated with insulin resistance. And they typed every participant in the study for these SNPs. So whether they had the risk allele, right, or one of the other alleles, and they gave them a score, and they called this the 53 SNP score. And this was used as a continuous variable. They also did functional analyses on 3 of the genes that were thought to be the driver for this relationship. And these were 3 genes that had never been identified previously as being involved with obesity and insulin resistance. And what they found is that the 3 I've listed here were involved in adipocyte differentiation. So the bottom line, if you read this paper, is that if you can't store fat, you become insulin resistant. The genes that were risk factors for insulin resistance were lipid storage genes. And if you had the risk allele for those and you couldn't store fat, you become insulin resistant. So this is exactly opposite of what we've always been taught, right? We're always told, oh, you become obese first, and then you become insulin resistant. And now what I'm saying here is that it's the people who can't store fat who become insulin resistant, which makes you scratch your head. But actually, we already knew this, because if you think of lipodystrophy, which is a pathologic genetic syndrome, these are folks who can't store fat, and they're severely insulin resistant. They're also prone to type 2 diabetes. They have liver fat. They have triglycerides in the thousands. So not being able to store fat is a very bad thing, right? Humans are actually designed to store fat. It's okay if we store fat. We wouldn't be able to survive if we didn't. But the new way of thinking about this now is that if you can't store fat, you become insulin resistant. So let's take a look at the actual data from this study. So what we're looking at here are these forest plots. On the left over here, what we have is the anthropometrics. So people who have the higher risk score statistically have a lower BMI. That's right. They have a lower BMI. They also have a lower percent body fat, but they have a higher waist-hip ratio. But their waist is not higher. It's their hips that are smaller. So they have small hips. This is the hip and thigh region, which is where humans deposit most of their fat. So they have this deficit in fat in the hip and thigh area. They have a high fasting plasma glucose, a high 2-hour glucose, and a high A1C. Over here, we have the DEXA and CT scan data. And what jumps out at you right away are these 2 bars right here. This is, if anyone's ever done a DEXA for body composition, you can get gynoid fat, which is the hip and thigh region, and you can get leg fat. And you see that these are both really low in the individuals that have the elevated risk score. What's interesting is if you look up here at visceral fat, statistically, it's not greater, although it does tend to be a little bit higher. So what's really going on with these people is that they can't store fat in their legs. And they have prospective data, longitudinal data, looking at incident type 2 diabetes and incident cardiovascular disease on these participants. And what we're looking at here is incident type 2 diabetes on the right, plotted against looking at the same time as leg fat over here. So this is by quintile of leg fat. So this quintile here has the lowest leg fat. And then this is the same quintile that has an elevated risk of incident type 2 diabetes. So lower leg fat, greater risk for incident type 2 diabetes. But I want to point out a very important point, is that statistically this interacted with weight gain. the incident diabetes was exacerbated by weight gain or positive energy balance. So not obesity, but weight gain. This is the active process of gaining weight. So they gained weight over the time course of the study. And the individuals who had the risk score and gained weight were actually the ones who got type 2 diabetes. So what this looks like now is that you have this genetic predisposition at birth to impaired peripheral adipose tissue expansion ability. And as a result, you have a low BMI. Now, what was not part of this study, but was implicit in understanding and interpreting the results, is that you would presumably have elevated ectopic lipid. And this is especially true if you gain weight, because if you gain weight and you can't expand your adipose stores, where are you going to put it? Well, you're going to put it someplace it doesn't belong, like the liver or inside the muscle, the heart, the pancreas, and so forth. It's going to go somewhere it doesn't belong, and it's going to cause trouble. But that piece of the puzzle was not available from the UK Biobank and the associated studies. But presumably this at the molecular level is ultimately what causes insulin resistance, which in turn causes chronic disease. So this is very oversimplified, but you can think that there's two obesity phenotypes then. You have this insulin resistant phenotype that has poor adipocyte differentiation, low peripheral subcutaneous adipose tissue, greater risk for chronic disease, relatively low BMI. So this is the profile that you see, the relatively thin person with the visceral fat. On the other hand, your insulin sensitive phenotype actually is your healthy obese. Which you've heard more about in recent years. This is this very controversial new concept, is there's actually this healthy obese phenotype. These are individuals that have greater peripheral adipose tissue, for example, in the legs, relatively high BMI, but lower risk for chronic disease. And this seems like a fairly artificial and arbitrary dichotomy, but these two phenotypes are actually really, really different. And this is particularly apparent if you provide them with a metabolic challenge and you look and see how they respond. And this is my favorite study that did exactly that. This is out of Sam Klein's lab at Washington University in St. Louis. And what they did is they recruited a group of obese individuals and they dichotomized them into two groups: metabolically normal MNO and metabolically abnormal MAO. So the metabolically normal are the healthy obese, and they overfed them. And what we're looking at here are the data at baseline and after overfeeding in the two groups. So what we have in the upper left here is glucose rate of appearance during a euglycemic clamp. So this is hepatic insulin resistance. So lower is better. Okay, this is hepatic insulin resistance. And if you look just at the white bars, which is before overfeeding, you see that the metabolically abnormal are more insulin resistant at baseline. And in response to overfeeding, they get worse. But what's interesting is the metabolically normal, they handle the overfeeding just fine. Their hepatic insulin resistance doesn't change. And then over here on the right, we have glucose rate of disposal during the clamp. So this is skeletal muscle insulin sensitivity. So higher is better. And if we look at baseline, the metabolically normal have higher insulin sensitivity than do the metabolically abnormal. And it doesn't change with overfeeding. Whereas the metabolically abnormal show a significant decline with overfeeding. Down here, we have VLDL ApoB100 secretion. You see the same pattern. The metabolically abnormal show an increase with overfeeding. But what I think are perhaps the most interesting data of all are this last panel here on the lower left. This is gene expression in adipose tissue before and after overfeeding. And what they're looking at are some elongases and desaturases and enzymes that are related to the ability of the adipose tissue to expand and take up and store lipid. And what you can see, this is the metabolically normal here. With the overfeeding, they show an increase in gene expression of these various enzymes, whereas the metabolically normal show no change. So at the molecular level, the metabolically normal can accommodate overfeeding. And you can see, I mean, I think it's really striking that you don't see changes in insulin resistance or insulin sensitivity with overfeeding. This just completely goes against everything we've always heard. And that's because the data sort of gets swamped out by when you mix the metabolically abnormal and the normal, you're going to see a decrease in insulin sensitivity because of the metabolically abnormal, but it's two completely different groups of obese individuals. So risk for type 2 diabetes is conferred by weight gain. Again, I use it, it's an active term. It's this positive energy balance, which stresses the system. People who are obese are actually fine in a sense because they have their fuel stored away safely for a rainy day. But weight gain on a susceptible genetic background, this gene by environment interaction, is what will tip the susceptible individuals over the edge. So our updated etiology of metabolic disease then is actually like you're dealt the cards you're dealt at birth. So again, I'm sure it's oversimplified, but it's a use just for the purposes of conceptualizing it. You could be born an insulin resistant baby or an insulin sensitive baby, which they probably don't look any different phenotypically at birth or even during childhood, but with age, they're going to develop into a little bit more of a physical phenotype that you can recognize, and particularly with weight gain. So the insulin resistant baby gets the apple type shape and the insulin sensitive baby gets the pear type shape. But they're all okay until they gain weight. And it's with weight gain then that you tip this phenotype, the insulin resistant phenotype, over the edge, and they end up with chronic disease, albeit at a lower BMI. And again, we are presuming that this is driven by the deposition of ectopic adipose tissue. So everything I've showed you so far is built on data primarily from, at least with the European studies, from European participants. And I think UK Biobank and those studies had largely white European participants. And what I'm wondering is how this translates to Birmingham, Alabama. Do we see similar things going on here? So we did a study. It was with 3 different ethnic groups: European American, African American, and Hispanic American. It turns out these are actually children. We did this years ago. It was trying to understand more about why ethnic minorities are at risk for chronic disease. And so we measured insulin sensitivity in all the children using an intravenous glucose tolerance test, which is actually not what the European study was based on. If you remember, that was fasting insulin. So we really gambled when we did this because this was a big leap, and this was children compared to adults. This was the United States compared to Europe, and it was an IVGTT compared to fasting insulin. But we wanted to see if this 53 SNP score worked. Essentially, even if we saw a signal. This is also a fairly small sample size. It's only around 300. And so what we're looking at here is that the 53 SNP score, which is what we call the genetic risk score here, versus leg fat from DEXA and versus insulin sensitivity from IVGTT. And what you see is we do in fact get the predicted inverse association. This was statistically significant overall with insulin sensitivity, and it trended in the right direction with leg fat. But what was interesting is when we broke it down, we reran it by ethnic group. So we did the same thing within the European American, African American, Hispanic American kids. And we found that both relationships were highly statistically significant in the Hispanic American children only. Although we did, with the African Americans, we got the same trend. If you look at the white children, which is the dotted line, in some cases that looks like it's doing the opposite thing. But in the Hispanics and African Americans, we got the same trend, and it was statistically significant in the Hispanics. Over here, what we're looking at is leg fat mass from DEXA, and it was different in all the groups. So Hispanic Americans had the lowest, which it fits with the fact that they had a higher risk score too. Statistically, the Hispanic American kids had a higher risk score, and they had statistically lower leg fat. The African Americans actually had higher leg fat than the Caucasians that were in the middle, and we'll come back to that later. So what this is saying is we know that type 2 diabetes risk is very high in Hispanic American individuals. It's quite high. They're one of the highest at-risk groups. And what this is saying is that there may be a genetic basis for this, and it may have to do with impaired peripheral adipose tissue expansion or storage capacity. And this is interesting because it turns out that Hispanics have been known for a number of years to have another genetic risk score for type 2 diabetes. And this is the Pnpla3 mutation. This is a gene, interestingly enough, that's also involved in lipid storage. This is primarily relevant in the liver. So the liver, the hepatocytes store these droplets of lipid, and which then have to be mobilized when necessary. And this Pnpla3 is involved in the mobilization. So if you have the isoleucine to methionine mutation, you can't mobilize. So it's like a one-way street. The lipid comes in and it can't go out. So people who are homozygous for the methionine mutation are at very high risk for having fatty liver. Fatty liver, of course, is related to hepatic insulin resistance and type 2 diabetes. So what we're looking at here are data from the Dallas Heart Study, and there's two pieces of information on this graph. One is the hepatic triglyceride content, or fatty liver, percent fat fraction, and the other is the frequency of the mutations in the various groups. And again, this is a tri-ethnic study, European Americans, African Americans, and Hispanics. So if you look first at the frequency of the mutation, what you can see is 99 of the Hispanics were homozygous for the mutant allele, and there was only about, 300 maybe Hispanics, so a fairly high frequency, but only 18 African Americans out of over 1,000. So much lower in the African Americans, and only 28 European Americans out of about 700. So Hispanics definitely have a higher frequency of the mutation. But then if we look at hepatic triglyceride content, what you see is that in all of the groups, the two that have the homozygous mutation have higher liver fat. But this seems to be particularly true in the Hispanics. It seems that the Hispanics have higher liver fat, even regardless of the PNPLA3 mutation. And this may have to do with the additive effect of their impaired storage capacity in peripheral adipose tissue. So the interesting thing about PNPLA3 is that it responds to diet. I think there might be an insulin response element in the promoter. I'm not 100% sure of that, but what I do know is that PNPLA3 responds to glucose and insulin. It's profound and it's very well established. So this particular study was done only within Hispanic individuals. Again, this happens to be children, and it's looking at the association between diet and liver fat. And the data are broken down by genotype. So this is the wild type genotype here. Here's the heterozygote, and here's the homozygote for the risk allele. And then we're looking at dietary carb intake and total sugar intake. And what you can see is that you get this striking positive association between total sugar intake and dietary carb intake and liver fat only in the homozygous mutant allele. So if you're Hispanic and you have the PNPLA3 gene mutation, to some extent, it's manageable. I mean, even though it confers a huge risk for liver fat, if you modify your diet, you could really minimize this risk. So this is like a brilliant example of precision nutrition that probably is not being practiced, but this seems to be low-hanging fruit. I mean, you could easily, if you can get people's genotype, you could counsel them not to eat excessive carbohydrates and definitely don't eat excessive added sugar. So what we know about the genetic basis for greater diabetes risk in Hispanics is that it seems to be related on one hand to this lower peripheral adipose expansion, which was the 53 SNP score, and on the other hand, to greater liver fat related to the PNPLA3 mutation. But again, this could be preventable, or you could minimize it. For one thing, you want to minimize weight gain, and for another thing, you want to minimize sugar and carbohydrate intake. So what we know so far about insulin resistance is that it's due to a genetic defect in the ability to expand peripheral adipose tissue. So the question then is the opposite true? We showed that if you have the one end, the high risk 53 SNP score, you have low BMI. But if you have the opposite end, if you have a really low risk, if you have a low 53 SNP score, do you have a high BMI? Well, obviously you do, because we saw it on the forest plot earlier. But this has never really been explored in terms of the physiological distribution of insulin sensitivity. Do more insulin sensitive people, are they at risk for weight gain? Well, we already know they are from a pharmacological perspective, because TZDs like pioglitazone invariably cause weight gain. And TZDs actually act on the PPAR gamma gene, which happens to be one of the genes in the 53 SNP score. So we kind of already know that insulin sensitivity causes weight gain, but at the same time, this information is not really applied. We don't think about it. We don't think about it when we're treating patients with obesity. Oh, are you insulin sensitive? And in fact, there's data out there. If you look for this in the literature, you'll find it. But my sense is that it's never really been given the attention it deserves. So we looked in our data to see if insulin sensitivity affected weight gain. And it turns out it does. And these are the data I'm showing you here. We did a study years ago. It was with women, and it was actually intended to look at obesity risk in African-American women. So we recruited a group of women that was half Black and half white. And these are women who had a personal and family history of overweight. So they were overweight when we recruited them. And we put them on a controlled diet for 16 weeks, and we got them down to a BMI of 25. And then we turned them loose. And under a free living condition, we followed them for several years. And we looked to see who gained weight, how much they gained, and what factors predicted weight gain. So what you're seeing are the 1 year data after weight loss. So everybody started out this study at a BMI of 25, and we're looking at their change in percent body fat over the next year. And it's broken down by two variables. I dichotomized it into low insulin sensitivity, which is abbreviated SI, and high insulin sensitivity. And I also broke it down into low glycemic load and high glycemic load diet. This is free living diet over that year. And I'll talk more about this later, but the reason I did it isn't intuitively obvious, because as we just talked about in the previous few slides, if you have issues with insulin, then you're going to respond to dietary glycemic load. So it seemed as though you couldn't really look at insulin sensitivity without considering diet. So what we can see here is that the group who showed the largest change in percent body fat is this group over on the end, who was high. But there was a very strong main effect of insulin sensitivity. Regardless of diet, the individuals who started with higher baseline insulin sensitivity gained more weight. But what was interesting is I mentioned that it was half Black and half white. I reran the same analyses broken down by race. So this is the African-American women on the left and the European-American women on the right. And you can see right away that the pattern looks identical as we just saw on the previous slide if you look at the Black women, but if you look at the white women, there's no pattern. Nothing at all was predictive of weight gain. So the whole issue with insulin sensitivity and diet was something that we were seeing only in the African Americans. So within the African Americans, we got a significant main effect of insulin sensitivity, and we got a diet by insulin sensitivity interaction. So women who had higher insulin sensitivity and ate the high glycemic load diet gained the most weight. So the question now became, what is it about being an African American woman that elicits this rather unique response to insulin sensitivity and diet? It turns out that African Americans have profoundly higher post-challenge insulin than Caucasians. Fasting is not necessarily different, but post-challenge is profoundly higher. And what you're looking at here are data from an intravenous glucose tolerance test. So what we do, this is glucose on the top panel, is we inject a bolus of glucose that's normalized for body surface area at fasting. So it goes way up. Within a minute or two, glucose is elevated and then it comes down. So what you're looking at is white and Black individuals. There's not a lot of difference in glucose, although statistically it's lower in African Americans. But let's jump down to the bottom now and look at insulin. This is the first phase peak. It's the area under the curve for the first 10 minutes. And the white participants, you can see it goes up to about 500 here on the y-axis, where it goes up to about 2,000 in the African Americans. And so it's fourfold higher. Ignore this peak. This is actually exogenous, but this is the physiologic response to that glucose load, is the area under the curve for this first 10 minutes. So what this is saying is that after a glucose injection, insulin in Blacks is fourfold higher than insulin in whites. We've seen this over and over and over. I've done it in children, young adults, older adults, men, women, and other laboratories across the country have repeated this. So this is not novel. This is solid as a rock. So now the question is, what causes this? Well, we still don't know exactly, but we have broken it down into whether it's secretion or clearance. And the answer is it's both. So here we're looking at the C-peptide data, which is secretion. It's purely secretion. This insulin is confounded by hepatic extraction. So when you look at peripheral insulin, the liver's already taken half of it, at least, away. So what we see here with C-peptide is how much is secreted from the beta cell, and it's twofold higher. So roughly twofold higher in blacks as in whites. So the differential then is hepatic extraction. So we've done a number of studies looking at clearance, and I'm citing one of the more recent ones here that was actually done with a newly developed mathematical model. And there is in fact about a 50% or twofold difference in clearance between blacks and whites. So this higher insulin that you see comes from two completely separate places. It's evolved, the beta cells seem to have evolved to respond more robustly to glucose, but at the same time, the liver extracts less. So it's kind of interesting from an evolutionary perspective, if you think about it. It's like the African Americans evolved to have high insulin. And there's two redundant mechanisms that have evolved. And you can, I mean, obviously we don't know why this is, but insulin's a very good storage hormone. So I think at some level, you could speculate that there's this thrifty gene hypothesis that's been around for years, and it may be something related to that. I mean, we'll never know, obviously, but all we know at this point is that in response to a uniform glucose challenge, insulin's profoundly higher in blacks versus whites. And this might be why the African-American women in this earlier slide here are so sensitive. If they're insulin sensitive and they have high insulin and they eat a high glycemic load diet, you have this very obesogenic metabolic environment. So I reanalyzed the data with this in mind, this high, we call it the acute insulin response to glucose, or AIR-G. That's this peak here. I reanalyzed the data, dichotomizing now by AIR and insulin sensitivity, low, low, high, high, and so forth. And you can see that it's just this group on the end that's high, high, that gains the most weight over 1 year. And in this model, there's no effect of race. So race completely drops out. You can replace race with AIR-G or vice versa. So the one is basically a proxy for another. So what we're seeing is that individuals with high insulin secretion and high insulin sensitivity are at risk for weight gain. And it turns out that we are not the only group to have discovered this. This paper was published in 1997, and it's looking at exactly the same thing. And I set this up the same way so you can compare them. But this was a study done over in the UK. It was just a free living observational study done in offspring of parents with type 2 diabetes. They recruited them at baseline, they did an intravenous glucose tolerance test, and they followed them for an average of 17 years. And they've expressed the data here as low, low, up to high, high, just like we did. And you can see they got the same thing. The group that had high insulin sensitivity and high acute insulin response gains more weight than anyone else. So I thought this was interesting and important. This is another group. These are offspring of parents with type 2. So this is another group that's at risk for metabolic disease, and they're showing the same pattern. So populations with high acute insulin response will be vulnerable to the obesogenic effects of high insulin sensitivity. And this, we know it's African-American women. Others have shown it's offspring of type 2 diabetes. But what about constitutionally lean individuals? There are obviously constitutionally lean African-American women. And we've done research that have included those women in our studies, and we have found that they also have the same high AIR-G. So how is it that, how do you dodge the bullet, essentially? If you have high AIR-G, how do you avoid gaining weight? Well, the answer is you become insulin resistant. So this is a study where we have two groups of women here. We have a group that we call obesity prone and a group that we call never overweight, or you can think of as constitutionally lean. The obesity prone were women who have a personal and family history of obesity, and they joined our study, and we put them on a diet and they lose weight. So in this slide, they're BMI 25. The never overweight are women who are never overweight, no personal or family history of overweight. And they were matched by age, race, and BMI to the obesity prone after weight loss. So everybody's BMI 25, actually less than 25. The range was around 23 to 25. Everybody in this graph has a normal BMI. And what we have on the left here are the European American women, and on the right, the African American women. So if you look at the European American women and you compare, this is insulin sensitivity, obesity prone and never overweight, there's no difference, although the insulin sensitivity is a little bit higher in lean women, which is what you would think. If you're lean, you have a high insulin sensitivity. But what's interesting, over in African American women, the never overweight, constitutionally lean women had a significantly lower insulin sensitivity. So what I think is going on here is that if you have high insulin, the only way you can avoid weight gain is to be relatively insulin resistant. And now what we don't know is if this has any risk for diabetes, but all I can tell you is statistically, these women have strikingly lower insulin sensitivity. So these are the data from the overweight women. The data, this is the same women here in the obesity, the two obesity prone groups. I just wanted to show you, this is before and after weight loss. So it's the same women. In the overweight condition, their insulin sensitivity was 3. And then when they lost weight, it went up to a 6. And the same was true in the Black women, started as a 3, went up to a 6. So there's no difference at all in the women who are obesity prone, their insulin sensitivity tracks. The group that's different is this one. It's these constitutionally lean African-American women who have this strikingly low insulin sensitivity. And I also wanted to point out, I know it's confusing because I'm talking about genetic insulin resistance. That is something you're born with, when we always think of obesity as causing insulin resistance. So they're not, those two are not incompatible. When these women gained weight, their insulin sensitivity dropped. I don't think I would call them insulin resistant, but your insulin sensitivity will go up and down depending on your body weight, but it's probably within a normal healthy range. So one more interesting thing about this group of women is the constitutionally lean women have very low insulin sensitivity. They have very high insulin. Metabolically, they also burn more fat. We put them in a 24-hour room calorimeter and we measured, this is non-sleeping RQ. So respiratory quotient is basically how much fat you burn. And the lower would be more fat. So lower on this one is better. Lower is more fat burning. And we used the non-sleeping because we wanted to include the postprandial period, which is when the women were eating and their insulin would be presumably going up. And we know that the never overweight African-American women have very, very high insulin, but which you would think would cause them to burn carbohydrates. But they're also relatively insulin resistant. And as you can see, this group here has the lowest RQ of all. So they're this group of never overweight Black women is burning more fat than everybody else. I just thought this was really, really interesting. So factors that affect weight gain, insulin response, or AIR, which what we've seen is basically a surrogate for race and vice versa, insulin sensitivity and diet. So it's a three-way interaction. And that's why I think trying to figure out what causes obesity is so hard, is it isn't just one thing. It isn't just what you eat. It's what you eat combined, superimposed on your metabolic phenotype. So how to intervene? Insulin sensitivity really can't be modified. It's probably genetic largely. But we do know that free-living glycemic load modulates the impact of insulin sensitivity. We saw that here. So the question now is, can you do this experimentally? If we recruit people with different metabolic phenotypes and feed them a low glycemic diet, what will happen? So we did this. We recruited a group of healthy, overweight, and obese men and women, Blacks and whites, and we put them on 2 different diets, a high glycemic load diet and a low glycemic load diet, and we followed them for 16 weeks. And what we found is that this is the loss of fat mass in kilograms, and the white bars are the white individuals, the gray bars are the Black individuals. And so the loss of fat over 16 weeks, if we look first at the the white individuals. There's no difference with diet. The hatch bar is the low glycemic. This is a low fat, high glycemic, and there's no diet effect. They lost a little bit more fat on the low glycemic, but not statistically different. So if you're white, it doesn't matter what you eat. Basically, this is what it says. However, if you're Black, it definitely matters. The African Americans on the low glycemic lost exactly the same amount of weight as the white individuals on the low glycemic. But the African Americans on the low fat, high glycemic lost only half as much. So again, I think this is another example of precision nutrition. If you know you have a high acute insulin response, if you're someone who secretes a lot of insulin, you're probably going to do a lot better on a low glycemic diet. And it turns out, once again, we aren't the only ones to do this type of work. There's a group up in Harvard that's doing the same type of thing. And I'm showing you here a paper they published in 2007 in JAMA. This is David Ludwig and colleagues. And he did something similar to what we did. It was a low versus high glycemic diet, but he did it in the free living. Ours was controlled feeding. He gave them a food list that told them what to eat. And what we're looking at over here, look at the far left first, is all participants combined over 18 months, looking at change from baseline kilograms body weight. Everybody lost weight and gained a little back, and it didn't matter what diet they were on. But he then broke them down into low insulin and high insulin. So, and he uses something similar to AIR. It's the 30-minute insulin from an OGTT. So these are the low insulin people. And again, it didn't really matter which diet they were on. They lost some weight, they gained some back. But over here are the high insulin people. Here, it did make a difference what diet they were on. The people who were on the low glycemic diet lost a lot more weight. And the interesting thing is they kept it off. Remember, this is free living. They were just given some instructional materials. They're given a packet of instructional materials. It was kind of like a 2-column list of foods. Eat this, don't eat this. And over 18 months, they kept that weight off. And when you see data like this, you know why these people struggle with their weight. When they're just told to cut out, and the interesting thing about this is I've seen the food lists. It truly is low glycemic. It's not low carb. They were eating bread. It's just they were eating whole grain bread instead of white bread and things like that. This diet would be super easy to follow with just a little bit of guidance on what you can eat and what you can't eat. And again, I think this is low-hanging fruit. This is extremely translatable if someone would just do it. If people would measure insulin at their doctor's office and instruct patients with high post-challenge insulin to eat a low glycemic diet, it would make a lot of people very happy. So the take home then, in terms of insulin sensitivity, is it can promote weight gain, but it interacts with diet and endogenous insulin. Obesity in African Americans is promoted by the high ARG, and it responds to carbohydrate restriction. So how does this relate to elevated risk for type 2 diabetes? Now, that's the interesting question, because really what I've told you so far is these are, the insulin sensitive people are the ones gaining weight. They actually may not be at risk. Right? We always sort of confound obesity with risk for diabetes, but what we know here is that the insulin-sensitive individuals are gaining weight. So the whole question of how this relates to diabetes is interesting and I think unresolved. And I have a couple slides at the end here I want to share with you that address what we've been doing to try to chip away at this. So this is a study we did using the euglycemic clamp. This is with adult African Americans and Caucasians. And I just wanted to show you the relationship between body mass index and insulin sensitivity. It's inverse, as you would expect, but as you can see, it differs dramatically in whites and blacks. So the whites are the open and the blacks are the filled. And there's two things readily apparent from this slide. One is that the slopes of these two lines differ dramatically. It's much steeper in the white participants. And the other is that the variance varies dramatically. So if you look at the line of the white participants, these dots line up like birds on a telephone wire. But if you look at the variance in the African Americans, it's quite a bit more. And we already kind of know why that is. It's because we saw previously on earlier slides that the lean individuals who are down at this end are genetically insulin resistant. These are your constitutionally lean African Americans, whereas anyone who was insulin sensitive gained weight and moved up this line, and now they're up here. So this is very dynamic down here, and there's different things going on. I don't think that's true in whites. We have no evidence that that's true. I think with the Caucasian participants, they gain some weight, their insulin sensitivity drops, and they just drop down this line a little bit. But there's a significant interaction here, obviously, and I think it tells you that there's two very different things going on. So BMI doesn't explain insulin sensitivity in African Americans, but what does? Well, we already alluded to this earlier when we looked at the kids study, and I showed you that the thigh fat was higher in the African Americans, and it was related to the genetic risk score. So now we're looking at the data from this is a population of adults here. We're looking at multiple linear regression to see what explains insulin sensitivity. And it turns out it's fat distribution. It's leg fat and subcutaneous abdominal fat. And this explains 44% of the variance. So that the leg fat is positively related to insulin sensitivity. More leg fat, higher insulin sensitivity. But the abdominal fat is inversely related. So more abdominal fat, lower insulin sensitivity. But you don't see any of this unless you put them both into the model simultaneously and parse out the variance. They're both statistically significant. And again, we get almost half the variance in insulin sensitivity from body fat distribution, but not BMI. The other thing that explained variance in insulin sensitivity in African Americans was total lean mass, which is muscle and liver. But because this is glucose disposal, it's primarily, you can think about this as muscle. So total lean mass. This explained almost a quarter of the variance in insulin sensitivity in African Americans, but it was inverse. So higher muscle, lower insulin sensitivity. Not what you'd expect to hear, right? This was statistically significant. So why is higher muscle associated with lower insulin sensitivity in African Americans? We don't know, but we think we know. And I think it's because of fiber type. We did a biopsy on these participants, and the fiber type data are shown down here. Myofiber composition percent. If you look at type 1 fibers, these are higher in white participants, both men and women, than in black participants, both men and women. And then the type 2A fibers tended to be higher in the black participants. So there's a fiber type difference. And we know that fiber type affects glucose disposal. This is a study showing that type 1 fibers have much higher glucose handling capacity. So this is looking at protein. It's actually protein. These are Westerns, I believe. And they're looking at, they have type 1 fibers in the white and type 2 in the black from a number of different participants. And what you can see is that the type 1 fibers have a greater insulin receptor, GLUT4, hexokinase, glycogen synthase, and pyruvate dehydrogenase. These aren't all shown here. This is the hexokinase. You can see that this is really, really striking. The type 1 fibers have much, much more. So for any given amount of insulin stimulation, you're going to have a lot more glucose disposal with type 1 fibers. And that's what a clamp is all about, is you're looking at insulin-stimulated glucose disposal. And one more piece of this puzzle came when we looked at lipidomics. So we took another chunk of this biopsy and we ran lipidomics. And you probably have heard this story that diacylglycerols and ceramides are some of the active agents involved in insulin resistance. So at a molecular level, we know from a lot of mouse studies, from a molecular level, that diacylglycerols and ceramides impair insulin signaling, and they've pinpointed exactly at what step this occurs. But the point is, if you accumulate lipid in your muscles and you don't oxidize it, and it accumulates and it degrades into toxic intermediates, these can impair insulin signaling. And the two culprits are the diacylglycerols and the ceramides. So we looked at diacylglycerols and ceramides. We also looked at a lot of other stuff. So I'm just showing you a little piece of this. And we looked at their association with insulin sensitivity in African Americans and Caucasians. So on non-Hispanic whites here, this side of the plot is labeled inversely associated with insulin sensitivity. And here's total diacylglycerols, and here's a bunch of the species, and here's ceramides. And you can see that these are all inversely associated with insulin sensitivity. But look at the right-hand side. These same species, the diacylglycerols and the ceramides, are positively associated with insulin sensitivity in African Americans. So completely opposite relationship. And once again, this can be explained by fiber type. There are animal models that have, humans tend to have heterogeneous muscles in terms of fiber type, but some mice have very homogeneous. You can pull out a type 1 muscle. And when you do that, you find this non-Hispanic white pattern over here. But if you pull out a type 2 muscle, you get this. So type 2 muscles show this positive association between diacylglycerols and ceramides and insulin sensitivities. So this supports what we suggested on the previous slide, that this inverse association between skeletal muscle and insulin sensitivity may actually be an issue of fiber type. So in a sense, I'm not sure, it's an artifact of fiber type, if you want to put it that way. And I don't know that it's pathologic in terms of risk for type 2 diabetes. So the whole issue of whether insulin sensitivity has anything to do with type 2 diabetes in African Americans, I think this brings this very much into question. So if that's the case, why do African Americans have a higher incidence of type 2 diabetes and prevalence? And obviously we don't know. And what we're looking at now are the beta cells, because that's the other piece of the picture. In order to become diabetic, you have to essentially have beta cell failure. Insulin resistance definitely plays a role, but the beta cells kind of play the final pivotal role. If your beta cells hang in there, you're never going to get diabetes. So we looked at several measures of beta cell function, and this one is proinsulin. Proinsulin is the precursor molecule to insulin. It's usually processed into insulin and C-peptide before secretion, but everybody still secretes a little bit of proinsulin. But the idea is that you don't really want to, and more is worse. So what we see is that it predicts impaired glucose tolerance in type 2. You can predict conversion to type 2 by proinsulin levels. And there's actually a group over in Germany who's developing a point of care test for this because it has such powerful predictive ability. But basically, it's a measure of beta cell function, higher being worse. And we measured proinsulin normalized for C-peptide or total insulin secretion, both in the fasting condition and after a 2-hour glucose challenge in blacks and whites. And we found in both time points it was higher in African Americans. And again, these are healthy, non-diabetic. That was our recruitment criteria, fairly young adults. And statistically higher proinsulin in African Americans. And further, this was inversely associated with the Disposition Index. The Disposition Index it's kind of a global measure of insulin action. It's the product of insulin sensitivity and beta cell function. We're looking at the dynamic phase or first phase, which is the one that's important for risk for diabetes. And this is at 2 hours, but we also saw it at fasting. We see an inverse association between proinsulin levels and the Disposition Index. So it's suggestive that this elevated proinsulin may be associated with poor beta cell function. So the basis for greater diabetes risk in African American, maybe we should be looking more at beta cell function than insulin resistance. And we don't know why the African Americans seem to be showing impaired beta cell function, but my speculation is it's due to this high AirG, which we see in healthy young children. So if you think of a lifetime of that excessive high insulin secretion, it may eventually wear away on the beta cells. However, beta cell function is exacerbated by insulin resistance, which we have seen in African Americans, particularly the lean phenotype and obesity. Obesity independently is associated with stress on the beta cells. It's associated with greater beta cell responsiveness, even independent of insulin sensitivity. So anytime you see obesity, it's a stress on the beta cells. So prevention in the case of African Americans should focus on minimizing, again, sugar carbohydrate intake, which will stress the beta cells. But we've also seen that low glycemic diets will minimize weight gain. So you kind of get both benefits from eating a low glycemic diet. And before I close, I just want to acknowledge support from Niddk, which has supported everything I've just shown you. And our core facilities at UAB, we have the CCTS, the DRC, and the NORC, which provide the infrastructure for all the measurements we do. Many postdoctoral fellows, collaborators, and students have participated in the research I've shown you. And thank you for your kind attention, and I'll take any questions.
Thank you so much for that. Definitely comprehensive, and I always find it informative. So there are a few questions in the chat that I'll start with. If you want to ask your question, please just turn on your camera. I did have the first question in there, going back to one of your first slides, talking about that large consortium study of the 300,000 patients where you showed the lower hip. Presumably that was male and female combined. Is that same hip to waist ratio true for both sexes?
There's a, as I'm sure you know, men have a higher waist and smaller hips than women, especially premenopausal. This changes a little bit after menopause. So that those data I showed you were definitely everybody lumped together, and they probably, I'm sure they adjusted for sex. Men do deposit fat in their hips and thighs. You know, I think in everybody, no matter who you are, that's where most of our fat gets deposited. We tend to think of it as the female fat pattern, but men have it too.
Okay. Liz, did you want to expand upon that? You kind of answered her question, but.
Yeah, I was asking about postmenopausal, whether it held true, whether it was different. You already answered it. Thank you.
Okay, I'll skip my second one. Let Pons get a chance.
Yeah, I mean, my question is, which among these two types, that is defect in expanding peripheral adipose tissue or greater adipose tissue accumulation, is high risk for cancer, and why? Or both?
Yeah, so I think the whole field of metabolism and cancer, I think, is fascinating, and I think it's underexplored. And I know that UAB Cancer Center is hiring right now. They have an ad posted for a metabolism cancer slot, in part, along with several others. But I think what we know from a lot of the data are epidemiologic, and there are data pinpointing insulin, high insulin associated with higher incident cancer. Now, one thing to remember is that all the cancers, you can't really lump them. So breast cancer, for example, is very sensitive to insulin. GBM is very sensitive to glucose. So I think there's a link between metabolism and cancer, but I think you have to be a little bit careful about looking at the specific metabolism of the specific cancers. However, I think from a global sense, the handwriting's on the wall is that the poor metabolic health that's putting people at risk for heart disease and diabetes is also putting them at risk for cancer. That's the sort of the big picture. What we don't know is the little picture. We don't really know. We don't know the answer to your question is if peripheral adipose expansion has anything to do with cancer risk. I would guess it does. It would be a really good study to do, and there may be a way to do it using existing data sets. I think there's a lot of work to be done in that area, and I think it's important.
Thank you.
Shuko, are you still on?
Yes.
Okay. Do you want to ask your question or?
Sure. Yeah. So it really interesting, fascinating that changing my assumption about insulin resistance and obesity. My question is like, if there's any data on Asian ethnicity in terms of this, and then also healthy obese, it seems to be okay to be for the diabetes, but it does increase the risk for other things like cardiovascular disease or something, atherosclerosis or something like that.
So the healthy obese issue is really controversial. And there's going to be people who argue there is no such thing. You can't be healthy obese. And I think it's a matter of degree. I think at some point you can be, but there's a limit, and anybody can gain enough weight to go above that limit. And that's why, it's always important to take a look at your triglycerides and your glucose. Those are your easy indicators of whether you're okay or not. And I did not hear, so in terms of other diseases, yeah, most of this has been done regarding diabetes and probably cardiovascular disease, and not so many other chronic diseases. So a lot of this we just don't know the answer to. But diabetes and heart disease, I think diabetes is the biggest single risk factor for cardiovascular disease. I think those two are really, they have a lot of common underpinnings. So I wouldn't be a bit surprised if a lot of what I told you today doesn't apply to cardiovascular disease also. And I did not hear, Asian ethnicity. Yes, there are data. There's lots of them. I don't know them as well because we don't tend to have the populations here in Birmingham and I don't study them. But what I can tell you is that it's different. They're all different. In terms of insulin resistance and beta cell function, there's a completely different phenotype with Asian ethnicity. And obviously, as I'm sure you know, the BMIs are smaller and the risk factors occur at lower BMI. And I don't know much about fat distribution and ectopic fat in Asian populations.
Okay, Great.
Thank you.
Gresh, can you ask our last question?
Sure. Really wonderful talk. I enjoy it because some of my lab also work on that area, especially obesity in reference to cardiac disease, muscle disease, and particularly in reference to circadian clock. So you mentioned about many times in your statement, you said like insulin is genetically regulated, but I think many studies say that it is also regulated by circadian clock. So can we say it's completely genetic? How much our environmental can change that? And that was like on your statement. So that's what I want to talk about. But I think I have too many questions, like maybe you can set up another meeting, but it was really wonderful talk. Let's get with this. And then I have one more question right now.
Thank you. And I know the circadian clock definitely it interacts with metabolism, there's no question. So my talk was very focused on a specific topic, and there's lots of other variables that are going to play into insulin resistance, beta cell function, and so forth. And the circadian system obviously is one of them. I think that people who, your night shift workers and people who don't get enough sleep, that has been meticulously examined in terms both of insulin resistance and beta cell function. And that's just another environmental insult that's going to superimpose on top of everything else.
Yeah. So my other one, because in several studies you mentioned about their food time, all that on. So how much that is like affecting your study? I think for some study you mentioned, we also mentioned the food, how much they are when they are taking that food. I think the timing of the food. If I remember correctly, you said the timing of the food for one of your study. And so what I want is we do that feeding fasting rhythm regularly. And one of the pathway, Gene, you mentioned that triglyceride, that was also influenced by our transcriptomic analysis, and we followed that at genetic level. And the paper is under review right now. I can talk more about that in near future, but that was really wonderful to see that in the obese people as well as in our basic model like fruit fly model that mainly I work with. So how much is that the feeding fasting, or especially the food timing you take into consideration when you are doing those wonderful human population-based study?
We don't do much with timing other than that when we feed our participants, we tell them, we give them their breakfast, we give them their lunch, we give them their dinner, we give them their snacks. And we try to enforce that they eat these things, but we don't tell them when to eat them. And most of the time, we probably don't even consider when they eat them. So some of our patients are late eaters. They sleep in, they get up, they eat their breakfast later. It doesn't come into play. I think it's an interesting question. I think especially when you're dealing with participants with diabetes, there's been some studies done showing that what they eat and when they eat it can make a difference. But so far we have not played around with that. It's interesting.
So my
Well,
last
thank you again. In respect of everybody's time, we're a little bit past the hour, and we can
That's
definitely
fine.
continue that, but I know some people have other meetings that they have to get to. Definitely thought-provoking. I have still a list of questions I'll be emailing you. So thank you so much for that. I appreciate it.
Okay, thank you all. I enjoyed it.